Greg, for that erudite and unflattering introduction. It's a real pleasure and honour to be here. I'm delighted to be delivering this year's Stanley Home Memorial Lecture. I'm delighted that you've invited me here to this terrific university. We've just learnt of its expansion and its meteoric rise up the performance tables, Times Higher Educational performance tables, and I'm thoroughly impressed by the past 15 years, and I can see on its current trajectory we won't have to wait another 15 years before we're biting at Oxford heels. Okay, so before I start my lecture, you'll notice that on the arm of your chair there is a chocolate. So I was about to say, please don't eat it yet. This is my gift to you, and there will come a time when I'll ask you to unwrap it. But, and I do have a spare if you want. If anybody else wants a spare, then there are some. Okay, so I'm going to talk this afternoon about something. I'll give you a kind of a light-hearted introduction to a very serious subject, and something that I've been working on for many years, but has just reached fruition in as much as that we've just begun to do clinical trials in this area. Before I do that, I want to just remind you how, using two statistics, how important the field of cancer research is. So this is one statistic that really shocked me. I learned it 2 or 3 years ago, that now cancer has overtaken heart disease as the main source of morbidity and mortality amongst the under 85s. And when you dig down into the numbers, you see that the demographics are even more striking. So the age of diagnosis is getting earlier and earlier for some cancers, not all, but some major killers, including colorectal cancer. The second startling statistic is that if you look at the projected incidence of cancer over the next 40 to 50 years, what we're going to see is a doubling to trebling of the number of cases globally. That's partly driven by global population, but it's also driven by other etiological factors. And it is not getting better. So, for example, by the year 2070, we can expect to see 3 times the number of cases of colorectal cancer that we do today. Now, that's an unsustainable burden. You're setting up a new medical school here. You won't be able to train 3 times the number of oncologists to deal with that number of new diseases in 45 years. And it's a terrible disease burden. So there is an increasing pressure on us all as cancer researchers to focus on prevention rather than just therapy. So how are we going to do that? Well, a clue for me came relatively recently in the trajectory of cancer research, about 15 years ago, with the deployment of immunotherapy. So this is ways in which we can augment our own immune system to attack cancer and reduce cancer burden. It's a completely new kind of cancer therapy. It's a so-called living drug. Your immune system evolved to attack infectious diseases, but it may be possible to turn the same mechanisms onto attacking cancer. Because it's a natural process, a natural system, it can also evolve with the disease. Evolve in a positive way. So as a cancer finds ways of escaping a therapy, your immune system can adapt and track those changes and be equally effective years hence. And critically, immunotherapy has been shown to cure cancers. Now, you'd never, 20 years ago, hear an oncologist use the word cure, but around 10 years ago, that began to creep into the oncology lexicon as a result of clinical trials that started about 15 years ago. A long-term follow-up has shown that there are now complete remissions of otherwise lethal cancers. And that, of course, has led to an explosion in the number of clinical trials focusing on immune targeting. These are the numbers from 2018 through to 2022. There was a COVID-induced blip around then. These numbers have picked up, and you'll see 2026 numbers out here at about 4,000 clinical trials per year. When you look at the kind of clinical trials that are out there being conducted right now, you'll see that the vast majority of them target T cells. I'm going to tell you what T cells are in a moment. And many others, most of the rest, are so-called cell therapies. These are also a kind of T cell therapies. Okay, so what are T cells, and why are they so important to us as we investigate new ways of treating cancer? Well, if you look deep into a tumor, this is a small but lethal, potentially lethal melanoma, you'll see that there are not only cancer cells in this mass, but there are lymphocytes. And when you look at the kinds of lymphocytes in there, they're these so-called cytotoxic T cells. And these are recognizing tumor cells, and their function is to secrete toxins that kill the cancer cells. These cells evolved to attack virus-infected cells, but it turns out that they can also recognize cancer cells. And I've spent most of my career figuring out what they can recognize. So we do know, let me just show you some experimental data, it's observational data, that shows that the more of these cytotoxic T cells that you've got, you can identify in a tumor mass. These are tumor samples. These are all the cancer cells in that mass. And the brown cells here are cytotoxic T cells. You can see that this whole mass is infiltrated with cytotoxic T cells. And you can use the number of cytotoxic T cells to stratify patients into outcome probabilities. So if you just look at cumulative survival of a cohort of patients, and this was actually a study that we did in head and neck cancer, you can see that those with high numbers of TIL, these are tumor-infiltrating lymphocytes, have a good prognosis, but those with low levels of infiltrating lymphocytes have much poorer prognosis. So the T lymphocytes are doing something positive. And this is natural. This is not in response to any therapy at all. This is just an observation. Okay, so I'm going to take you right back to basics. It's rather embarrassing that we've got maybe faculty in the audience, we've got school children, but just bear with me. So what do T cells recognize? Well, we all know that inside the nucleus of cells, our genome is arranged into genes, and each gene encodes all the information to make a protein. These could be structural proteins like myosin in muscles, or an enzyme like insulin that regulates your blood sugar levels. But that's how we build normal life. But our genes, which are made of DNA, are under constant attack from all sorts of sources, including environmental carcinogens, ultraviolet light, for example, natural cell processes, so natural mistakes that aren't corrected. Some inheritance can introduce mistakes that are inherited through generations. And viruses can also disrupt the structure of genes and the sequence of them. And the upshot of many of those processes is the synthesis of an abnormal protein. So this is a protein with a specific change in there. It may be one amino acid amongst hundreds. And the protein may be made and perform an abnormal function. Okay, so this is where the chocolate comes in. So pay attention from now on. So how are these changes recognized by T cells? Well, those proteins, the normal ones and the abnormal ones, have to be processed. They're turned over as a normal part of life. They're made and they're degraded. So if you take up your chocolate now, so what you're holding in your hand now is a copy of a cellular protein. It could be a protein that's involved in normal cellular cell regulation like p53. Okay. So what happens is that p53 is turned over. It's made, you hold it only in your hand, but then it's degraded, and that's all part of nature. So let's do the degradation. You unfold it, unwrap it, and you can process most of it, so you can eat the chocolate if you want. But the important thing is with the wrapper, just hold it above your head and wave it. Just the wrapper. Okay. So I now am a T cell. Okay. Each one of you is a cell in the body, a normal epithelial cell. You've made a normal protein, and you're waving parts of that protein above your head. And you're using a molecule called a histocompatibility lymphocyte antigen, an HLA molecule. Okay? Now I'm a T cell, and my job is to survey you all and look for an abnormal protein. And what I see is if you wave your wrapper above your head, very good. I see lots of golden wrappers. These I recognize as normal cellular proteins. But as I scan the front row, I see a brown one. This is not normal. And so as a T cell, I can recognize you as being foreign, and I can come over and get rid of you. And so by doing that, I've prevented that abnormal protein from exercising its normal function and perhaps driving cancer. Okay, so that's what we've done. And so there are real molecules that do this. So the molecule that you represented with your hand is called an HLA molecule. And it looks like this. This is the real crystal structure of it. And my recognition process as a T cell is also a receptor on the surface of every single cytotoxic T cell in the body. And that has the capacity to recognize differences in normal proteins. So that's what we just did. Okay, and that's a process of molecular recognition that we worked out around 25 to 30 years ago. And I've now representing your gold wrapper as another kind of confectionery and M&M. And there's a good reason for that, as you'll see in a moment. I don't think this is working, this microphone, so if it's okay with everyone, I'm going to put it down. It is working. Okay, so that's the principle of molecular recognition of a tumor peptide MHC complex by a T cell receptor. Okay, so clearly our immune system has a way of identifying slight molecular changes in cancer cells and killing those cells to prevent them from propagating. So why do we get cancer? Well, I'm going to play another game with you now to illustrate how we can recognize cancer cells and why cancer wins under most conditions. I have another prop actually in my bag. So you've got to think of every cell in your body as being a jar of M&Ms. Every molecule is an M&M. They're all different colours. They perform different functions, and so on. So we can tell the difference between, on a macro level, between the tumour and the surrounding normal skin. But at the molecular level, they're much more similar than they are different. So that cell there, normal cell, looks very much like that tumour cell there when it comes down to the M&M level, the molecular level. Okay? So now you all have to imagine that your T cells, and you have to do what I just did and recognise the difference. So I'm going to put a picture of 15 cells, and one of which is different. When you've spotted the difference, I want you to put your hand up. You just put your hand up. Okay. We usually get one or two.
Use AI.
Okay, keep your hands up. So I think I saw three T cells recognizing the difference between the normal and the tumor cell there. Well, if that was real life, that's not a very strong T cell response. And so under those conditions, we saw a very weak T cell response, and the cancer wins out, and it carries on growing. Okay? So we're going to play the game again. Only this time, I'm going to give you all a clue. Okay? So we're going to play the game, and as soon as you see that difference, put your hand up. See what happens? So we get a really strong T cell response, and the cancer dies. Our immune system wins, and the cancer loses. And that's the principle of vaccination. It's a rehearsal of your immune system to recognize these differences, so that when they encounter those differences, there's a prompt and strong response, and we get rid of either the virus or, hopefully, cancer. Okay, so those molecular differences, this is a bit of science now, are called neoantigens. So they're mutations that can be potentially recognized by T cells, so we call them antigens. And if you look at different tumor types, you see that the number of neoantigens differs a lot. So what we've got up here is just the number of, I think they're coding somatic mutations per megabase of DNA. And they range enormously from virtually no genetic change in acute lymphoblastic lymphoma, right the way up to 10 to 100 nucleotide changes per megabase. This is a lot of mutations in melanoma, in lung cancer, lung, and upper GI cancers, and so on. And if you, since we've been talking about proteins, along the bottom here are the number of proteins that carry those mutations, and they range from virtually none through to around 300 to 500 different proteins carrying these mutations. And each one of those mutations could potentially be recognized by our immune system. And this is something that we would very much like to exploit in cancer research. Okay, so let me tell you a little bit more about how we accumulate these mutations. They don't all happen at once. They happen sequentially, and they can happen over a long period of time, 20 to 30 years in some cases. And they start in normal tissue, and they can accumulate as driving changes in that normal tissue through various stages of pre-cancer, which are all reversible, until some kind of crisis point happens around here, and so many mutations are accumulated that the cell regulation is so different to a normal cell that the uncontrolled cell growth and cell division results in a real invasive cancer. But it's not until multiple mutations have accumulated that this happens. And what I want to focus on is this part of the trajectory of tissue from it being normal, perhaps carrying one or two mutations, through to its stages just preceding the transition to cancer. So what happens, let's just think about how these mutations accumulate. You could represent these as sort of an evolutionary tree. So the very first mutational event down here results in all cells, can just be one or two cells, have this blue mutation here, and then as this clone develops, a cell within that expanding clone can accumulate a second mutation, a red one, but another clone can acquire a different one, an orange one, and so on and so on. So you build up this complex network of mutations, such that if you were to take a cross section through the tissue at this stage, this is the cancerous stage, you'd see multiple neoantigens, mutated proteins, some of which will be shared by all cells in the tumour, and others that would only appear in a few. Those that appeared more recently would only occur in a few tumour cells. So again, if we go back to our M&M analogy, some of those mutations, like the blue ones, would occur in all cells, others like the yellow in just some cells, and others like the orange ones in very few cells. So we've got a range of, we can classify our neoepitopes now according to when they were initiated in this developing tumor. We'll have those neoepitopes that are restricted to subregions, like the orange ones, because they occur late. And we'll have so-called truncal neoepitopes. They're called truncal because of this evolutionary tree arrangement that are widespread and are present in all cells in the tumour. Now, it turns out that many of these truncal neoepitopes are shared between patients. So if you look at tumours from different patients, they will all have a blue mutation. Okay? And critically, they more often than not occur in these pre-cancerous lesions. So these are tissue abnormalities that are not yet cancer in healthy people. And at the other end of the scale, we have a classification of neoepitopes that are regionally restricted. Most of these will be patient-specific. Okay, so they'll have occurred late in a disease, and that means that they'll differ between different individuals. And crucially, they only really occur in invasive cancer. So after the other side of this transformational episode that I've shown here with this sort of starburst. Okay, so this is where cancer vaccines comes in. So this is really the starting point for thinking about how we develop vaccines and how we personalize them to individuals. And you can see from this schema already that there are two avenues that we can pursue. The first is to make a vaccine to these to a cluster of neoepitopes that are specific for an individual tumor. And these can only be used as therapeutic agents because they really only occur in volume in tumors. But the other approach is to try and identify the blue mutations that are more widespread, so they work better in an individual tissue, and crucially, they occur in pre-cancerous lesions. So they may be driving the tumor, driving the tissue towards becoming a tumor, but they occur in very early lesions before that transformation event has been reached. Okay, and so that's exactly what is happening at the moment. So at this end of the spectrum, we have individualized vaccines. So these are the late occurring vaccines. There are clinical trials ongoing. There are some good success we're seeing from trials that BioNTech and Moderna are already running to identify personalized neoepitopes, make a vaccine, and use that therapeutically. And a second approach, which is the one that we've taken, and I'll talk a little bit more about it in a moment, are to identify those widespread, early occurring mutations that could nevertheless be immunogenic. These are fixed vaccines. We could use these across multiple individuals, and we could use them in populations that are healthy and have not yet had a diagnosis of cancer. Run through the process of making first a personalised vaccine. I like to describe this because even 3-4 years ago, this felt like science fiction to me, that we might be able to identify the neoepitopes in each individual tumour and make a vaccine in time to give back to a patient with a reasonable chance of having therapeutic effect. But that happened. So from a diagnostic biopsy, the whole tumor genome is sequenced, the mutations are called against the patient's own blood, so it's a very specific, so all the mutations that are called are specific to the tumor and not a natural variant occurring in the patient. We use some very flashy bioinformatic and now machine learning tools to predict which of those changes, and there are several thousand at this point, tens of thousands of potential mutations that we could target here. We have to narrow that down to a reasonable number of 20 to 30 to put into a vaccine. And so the algorithms that we've developed, based on our knowledge of the chocolate, which proteins will be processed and presented to T cells, the models now are pretty good at predicting which bits of a protein will be presented to T cells. We can use that information then to quickly manufacture a vaccine. There are multiple platforms available to us. I mean, thanks to one of the only good thing to come out of the COVID-19 pandemic was licensure of a whole new agent, mRNA vaccination. It had been an experimental drug for many, many years, but it was put into effect during the pandemic and proved to be a great success. That's now opened a door to a whole new way of making vaccines, and the field of cancer vaccination has been quick to adopt that technology, but it's not the only way, as I'll show you. Quickly make a vaccine in time to administer to the patient from whom you took the diagnostic biopsy, usually in combination with another immunomodulatory drug, which I don't have time to go into the mechanism of. And this is showing remarkable success. Now fairly large clinical trial, still fairly early phase. But this whole process now has been condensed by BioNTech into 4 weeks. This was a PhD thesis only 10 years ago, 3-4 years work. This is certainly an effective route to therapy. It's expensive, It's highly exclusive, partly because it's expensive, but access to the instrumentation, the expertise in the labs is highly exclusive. So we'll see whether it has a durable life. Instead, I want to talk about something at the other end of the spectrum, choosing shared early neoepitopes to vaccinate against. We've just completed the preclinical work on a vaccine to reduce the risk of lung cancer, which I'm going to describe in the next 5 minutes or so. So, the idea for a vaccine to reduce risk of developing invasive disease came from a piece of experimental medicine targeting a screening program for early detection of lung cancer. So there are several countries around the world that have now implemented these lung cancer health checks. The UK's rolled out last year, I think, or 2025, and came on the back of a summit study. It was which came out of University College London and the University of Manchester. So here, 2,500 individuals were targeted by their general practitioners. They had already had some risk factors for developing lung cancer. They were lifetime smokers. They were a certain age, so over 60, and they had a high BMI. So very generic risk factors, but very real nevertheless. They were invited to their local shopping centre, where the study team brought in a low-dose CT scanner, and just over 1,000 of them, or 1,400 of them, had this low-dose CT scan. Okay. Out of this, 46 of those 1,400 individuals had clear indication of early stage lung cancer. So these were they were healthy people. They didn't know they had cancer, but they were picked up. There's about a 3% detection rate of early stage cancer. And these individuals were fast-tracked into a curative surgery pathway. So they had surgery, and then we can't offer these people anything else. They do remain at high risk, so they come in for regular checks, but there's no further therapy scheduled for those individuals. Now, there's also another 13% of that cohort that had abnormal scans. So it wasn't cancer, but neither was it normal. There was some opacity or some nodule detected. And again, we can't really offer those individuals anything other than a regular screening program. But again, we felt that this group may also benefit from a targeted vaccine. Mm-hmm. So these are really healthy people. No signs of cancer. They've never had cancer before. And we'd like to target those with a vaccine. And we figure that with a cohort of around 750 of these individuals, we'd expect to see a 10 to 15% reduction in their risk from 30% down to around 20%. So we get a clinical signal within 3 to 5 years, which is a reasonable window for a clinical trial. So what did we do? So we identified 25 of those blue neoepitopes, and I have to follow this one through because I did it last night. I sorted all the blue ones out of our mixture here. So those are all our neoepitopes. So we identified all those out of this mix here. And we formulated them together with another cancer antigen that I've been working on for many years. And that gave us 96% coverage of all lung cancers, squamous and adenocarcinomas. So we knew that 96% of all patients have either one or more of the blue neoepitopes or this cancer antigen. And we put that together. We also, this might be a little technical, but we also predicted exactly which bits of each of those proteins would be presented to the immune systems of individuals with different HLA molecules. What I didn't say is that HLA is one of the most polymorphic molecules in the human genome. So we all have a different selection of HLA molecules. So we're all going to present slightly different pieces of chocolate wrapper. But we could predict, and we predicted over 17 different ancestries. Only 6 of them are shown here. Each blue tag here is a predicted response across multiple HLA. So you can see, all you need to see is that there is good coverage across multiple ancestries of predicted immune responses. And when we did that, we put all those neoepitopes into a vector that we could express in an adenovirus. So we used the ChAdOx platform. So this is the viral platform that formed the base of the Oxford COVID vaccine, the AstraZeneca vaccine. And we have, I'm very sort of proud of this slide. We've manufactured 1,000 doses of this already. They're already in the freezer. We have funding for the clinical trial provided by Cancer Research UK and Chris Cancer Foundation. And we're opening this trial in the first quarter of next year. So that brings me to the end of my lecture. I just wanted to conclude with the sort of key points that life is sweet because T cells can destroy cancer. So we do have the capacity, our immune system has the capacity to destroy tumors just as it has to destroy virus-infected cells. But vaccines work, but only if you can find the targets. If you can show your immune system what to recognize via a vaccine, then it will work. And I've shown you 2 different classes of target, those personalized neoepitopes and these shared neoepitopes, these blue neoepitopes that are shown here. And that's opened up 2 routes to the personal deployment of vaccines. So one way of personalizing vaccines is just to make a personal vaccine, so a vaccine that only works in one person. They do work, but they can only be used as therapeutics. And it's likely that they'll only work in combination with other immunotherapies. But the other route that I've shown you is to identify these shared neoepitopes and personalize them by focusing on risk factors, focusing on the right group to deploy these vaccines. I mean, I'm sure that in time there will be a pediatric vaccine to protect us against the lifetime development of some cancers. But for the time being, we have to think of it as a targeted approach. And for that, we have to identify the right groups in which to deploy these off-the-shelf vaccines. But the important thing is that we can now use them in a preventative setting. These are not preventative in the same way as you say, Rich, of preventing a viral initiator of cancer. These are preventative in a sense of being able to preempt what cellular changes are going to occur in a lifetime and vaccinate against those. And so please keep your eyes open to see what happens to LungVax in the future. And with that, I'll just thank the massive team that brought you LungVax. It's a collaboration between University College London, and the University of Oxford, with multiple funders, particularly funding for the clinical trial will come from Cancer Research UK, without which we couldn't really work out whether all this great science is going to make any effect at all.